The flu, also called influenza, is an extremely contagious respiratory illness. You hear reminders each year to get your flu shot, or once every 3 years if you're an animal. And although some vaccines provide lifelong protection with only one or a few doses, such as with the measles, mumps, or polio, the influenza virus requires a new shot every year. Why is influenza so different from other vaccines? Taking a look at the evolution of influenza virus can explain the weaknesses of current vaccines and can ultimately show us a way to provide universal protection. The influenza viruses are also called RNA genome viruses or retroviruses. Let's first talk about the replicative cycle of these retroviruses. The virus itself is made up of single RNA strands, reverse transcriptase, a viral envelope, a protein capsid, and glycoprotein on the outside of the capsid. This glycoprotein identifies with the other protein on the host cell so that the virus may enter the cell. After they use their reverse transcriptase to transform their single-stranded RNA to DNA, this viral DNA can then be integrated into the genome of the host cell. Then the host cell is tricked into copying these genes as part of its normal replicative process, so ultimately makes more of this virus again. First, these RNA viruses don't proofread as much as DNA viruses, so they reproduce less accurately. This results in RNA viruses having the highest mutation rate than any other organism on Earth. These mutations allow them to slip by the defense of common antibodies, produce more copies of itself, and eventually spreading to more victims. This process leads to the evolution of new strains, and some of which are likely to become the cause of next year's flu season. Secondly, influenza viruses are called the masters of reassortment. This occurs when two different viruses affect one host cell at a time. The viruses have 8 RNA segments, and they can swap genetic material with each other. Influenza virus has 8 RNA segments, with each carrying one or few genes. A popular research experiment from 2008 shows a human influenza virus and a bird influenza virus being mixed in a pig cell, since they are well known as mixing vessels. As you can see, the pig cell can release a new virus that contain a mixture of genetic variation from both the human and bird influenza virus. This kind of recombination is very common in nature. Action! The swine flu virus, also called the H1N1 strain, has RNA segments from birds, humans, and pigs from North America and Asia. This combo reflects a series of reassortment that occurred step by step over many years to produce the H1N1 strain. Now, you know the human's armors against retroviruses like influenza.